centers and it is an honor for us to have you all here. First of all, let me introduce myself. My name is Udayana. I'm from Faculty of Science and Fisheries, Udayana University. I will be the moderator in this session. Okay. First of all, okay, ladies and gentlemen, before we start the presentation session, I would like to inform you about the rules during the session. First, I will invite each presenter into the presentation for 10 minutes. The sequence of presentation will be conducted according to the list that has been given. We will help you to set up your PowerPoint slide, and the discussion session will begin after 6 presentation session. The allocation time for discussion session is 8 to 10 minutes. Okay, without further ado, we are going to start the presentation now. Please welcome our first presenter. Okay. Mrs. Tariko, yeah? From Udayana University. Thank you.
Okay, good morning everyone. First of all, thanks to the VIP of the room and moderator for the time that has been given to me. Let me introduce myself. My name is Tari Kosya. I'm a student at Udayana University in Aquatic Science Management major. So, in this occasion, I would like to deliver the results of our research with the title Preliminary Study Biodiversity of Collected Fishes consists of native and alien species in Batu Bali. Just for information, the research is collaboration of several foundations such as Udayana University, Universitas Nusantara, Muhammadiyah University, and Oxford University. Okay, as we know together, Batu Lake is the biggest lake of 4 lakes in Bali Island, and it has a lot of fish culture with multiple functions such as ornamental, consumption, and aquaculture. The purpose of this research are to knowing the biodiversity and ecological index in Batu Lake Bali, because the last research about fish diversity in Batu Lake Bali was conducted in 2011. The next is research method. The research was conducted on November to December 2021, and we used field process sampling method at 3 stations in Batu Lake covering Toya Bungka, Sakat, and Fisheries, as you can see on the screen. And the tools that we use are experimental units with varying sizes. Continue the previous, we analyze the data by 2 steps. The first step is fish identification. We identified the fish based on fish identification book by Mono and Margareta 2017 and fish-based website. And the second is ecological index covering biodiversity, uniformity, and dominance index. The formula that we use are on the screen, and the formula also will be used to classify whether the region has a high, moderate, or low pollution. Okay, we are going to the results. As you can see on the table, there are 516 individuals consisting of 4 orders, 4 families, and 12 species. Furthermore, from 12 species that we found, we identified there are 5 native species and 7 alien species. The most scattered fish are Amphilopus citrinellus, Barbonimus barbaroides, and Siphophorus celeri. As you can see on the picture, Barbonimus barbaroides come from native species, and we should maintain their population because it has an economic value. Usually, Barbonimus barbaroides used for consumption and ornamental fish because it has shiny color of the scales and nice skin. And the dominant by alien species actually can disturb and compensate the native species and sometimes cause an imbalance in ecosystem. So the efforts that we can do for decrease the population of alien species are by directing their population to the other utilizations, such as other promiscuity food we use as a main commodity in aquaculture in Bali, and other promiscuity food as the traditional food that usually called by Udayana. And by the way, most of species in Bali are fish with least concern status, but there is one of native species that including in vulnerable status, namely Rasbora latericiata. It's happened because Rasbora latericiata divided in competition for food by Orechromys nauticus and Orechromys echolithus. Okay, and the next result is about ecological index. As you can see on the table, Sekat water has biodiversity and renewability. I mean the highest value of biodiversity and renewability. It's because around the Sekat water, the activities are not very high. So the management of aquatic environment can be maintained. Otherwise, in Toya we found the lowest biodiversity and renewability because around the Toya waters, the activities are quite high, such as tourism and fisheries. In detail, there are hot spring baths and floating nature. Okay, and uniformity and Sekat water indicate that the communities were stable and the species compositions are even equally each one. And the last index is dominant. Actually, all locations have the low value, but the highest one is Toya water, with the most dominant species is Amphilophus citrinellus with 149 individuals. It's based on biological aspect of Amphilophus citrinellus. It has can breed very quickly, and it's predatory, which prey on the small fishes. And besides that, Amphilophus citrinellus didn't have an economic value, so the utilizations of this fish is low, and the population will increase all the time. Okay. One effort that can be made to decrease the population of Amphilophus citrinellus are by directing their utilization to fish, ornamental fish collection, and hopefully that activities can make Amphilophus citrinellus has an economic value. Okay, and last is conclusion. Fauna in Batu water consists of 4 orders, 4 families, and 12 species. And the most common fish are Amphilophus citrinellus, Barbadimus baleroides, and Scyphophorus celeri. One of the species in Batu that classified as vulnerable status is Rasbora latericiata. And the highest biodiversity and uniformity of this species was found in Sekat water. Meanwhile, the highest dominance are found in Toya water. And here are the acknowledgements. We use the leading research grant from Aquatic Resource Management Study Program, Faculty of Marine Science and Fisheries, Layanan University in 2021. And here are the references that we use. And I think enough for me. And thank you for listening and see you. Thank you very much. This is podcast for the presentation. Today we will next to the presenter. Mr. Dinyoman Diri Putra. And share.
Yeah, thank you for the opportunity. So in this occasion, I would like to present my research about the barcoding of cardinal fish in Gilimanuk Bay, Bali, Indonesia. The next slide. So, yeah, okay, thank you. So the Apogonidae is consists of nearly 300 fish species, but most of these species could not really differentiate by using their morphological character. This rise ambiguity to determine the species or the genre of the Apogonidae species. But in recent years, the coding methods is really developed, and we can use it to determine the species, the subgenera, and even to reveal the phylogenetic relationship of the species. But unfortunately, most of our barcoding research now in Indonesia is mainly focused on the commercially important fish, such as tunas or grouper, for the species that under conservation concern, like shark or rays. And less effort has been made for a small, low economic value and less conservation species. So this research aims to identify the Apogon species and establish the genetic database through the barcoding research using the CO1 of mitochondria. So the specimen collection, we conducted the specimen collection at the Gilimanuk Bay, located at the Gedung Agency, Bali. And the specimen collection was conducted at 3 sites with the various depth ranging from 2 to 7 meters. And we collected the specimen using a fish scoop net. And at the end of the survey, we successfully collected 22 samples of the cardinal fish in Gilimanuk Bay. And here are some photos and videos of our specimen collection. In this video, we can see some of the cardinal fish species, and one of them are Lantai cardinal fish. This one, this is the endemic species from the
Recording in progress.
Archipelago that introduced to Gilimanuk Bay. And that photo is when we're doing the sample collection, how we collect the samples and photograph and also preserve the samples in ethanol. Yeah, for the lab work, in order to get the sequence data, we develop a standard protocol for barcoding method. First, we extract the total DNA from the tissue using the Chilek 10%, and then we amplify the target locus of CO1 using 2 primer, BCL and BCA. And then at the end of the step, we define the nucleotide sequence by doing the DNA sequencing. And then here are some the data analysis that we are conducted. First, we edit sequence using the MegaX software, and then we doing blast to compare our sequence to the genetic database in the GenBank and TBI. And then we calculated some genetic distance, K2P distance in Mega also. And the last, we determine the phylogenetic relationship by construct a phylogenetic tree using RAXML. And to make the tree visualization more informative, we also use atau and Adobe Photoshop. And here's the result of our species identification. So all samples have a high similarity with the GenBank database, more than 95%, except for one sample code as Biosop 007, that according to the gene bank analysis, this species is identified as Australopithecus septemstriatus with the similarity of 87.27% and Australopithecus nigrofasciatus. But because the similarity is below the standard, the standard, so we observe or we compare the morphology of the species to the other two species that suggested by the gene bank. And here you can see there's some morphological characters that didn't match between our sample and the sample that suggested by the gene bank. So according to our morphology identification, we identify this species as Australopithecus hartfeldi. And this sequence is not really in the gene bank. So this research is found a new sequence of Australopithecus hartfeldi, and we already uploaded this sequence to the gene bank. So previous slide. Yeah, so based on blood and morphological comparison, we successfully identified 8 species, and this one sequence Australopithecus hartfeldi was new to the gene bank database. And then next slide. Yeah, this is the result of the phylogenetic tree. We construct the phylogenetic tree using maximum likelihood model, method with Hasegawa Kishinoyano model. And the result you can see here, each species is clustered into a distinct cluster within this species. So for example, this red one is the cluster of the Australopithecus hopeni, and the yellow one is cluster of Rhabdamia gracilis. So by using this mitochondrial DNA, we could distinguish each species of Apogonidae. Yeah, next slide. This is the conclusion. So the present study successfully identified 8 species of 7 genera of Cardinalfish, and 1 was new to the gene bank. And the barcoding method is effectively and reliably to delineate Apogonid species as the species level. Yeah, so I think that's all of my presentation. Thank you.
We have trouble with the Okay, we will continue to yeah, that's yours. Assalamualaikum warahmatullahi wabarakatuh. Let me introduce myself. My name is Udayana. I'm student in Erlangga University.
Can we have someone with the yang baru bisa yang berkaitan dengan dengan and let me start explain what my research. The paper of my research is about fish biodiversity and the correlation with start communication proposed for all the ecosystem in pengembangan budaya region. Yes. This start on pengembangan water. Jadi, divide into three types, provided with semi bottom and coral in 26 hectares and common cigarette in 9 hectares. Penimbangan is one of the potential locations of the underwater location for underwater tourism in Udayana Redaksi. But penimbangan has a unique best spot. In penimbangan, water has a planning and sustainable coastal development important for the community ecosystem and sea economy. Penimbangan has a charm from its support and development of special interest tourism activities. Penimbangan water area is a conservation area. Assessment of how effectively local coral can maintain how increased fish structure, fish biomass, and coral cover. Start monitor and maintain underwater in Penimbangan in 2060. Purpose of this research is examine the conditions of the reef structure and its relationship to the condition of the bottom cover in the water. This is Penimbangan water mine location research and divide into 4 sites. That one in depth 6.7 meter and yet site 2 in 7.4 meter, site 3 in 9.5 meter in depth, and site 4 in 11.1 meter. This research was conducted from December 2022 from 2023 at Lantai. Data collection was carried out in 5 different months to know whether there were different in water conditions or other environmental
sector. The method for the reef is use underwater photo transect, use quadrant bracket in 40 centimeter by 55 centimeter quadrat, and second by 50 frame photo. The observation area covered in 50 meter long. The underwater photo transect method advantage includes shortening in lifetime and provide estimate of the density of specific taxa with low methodological error. For the fish, I use underwater visual sensor. Underwater visual sensor is used right inside 2.5 meters and long 70 meters from start until end. The transect length is 70 meters long. Observation area all is 50 meters long. Underwater visual underwater visual method is fast, accurate, effective, and environmentally friendly. The resulting data is relevant to the objective of coral reef management and the general management of coral reef. The coral reef analysis is from 50 photos taken with coral point count software with Excel. Based on this table and criteria is bad, currently, good, and very well based on degree of Minister of Environment number 4 of 2001. Okay, this is the result of my percentage variation of sister covered. The highest is this coral with alga, and the next is coral, followed by sand. Category of life form coral is acropora and non-acropora. Acropora in 3 types, and the other is coral basin, until coral to coral. And the highest is coral massive, followed by encrusting and submersive. According to the Decree of Minister of Environment number 4 of 2001, the condition of coral reef in Pelimbangan is currently category with an assessment rank to 25 under 49.9%. For the next is the species of fish by family. Has a chepodon, Sanhilde, and Rabidae is found at its all types. Based on the type substrate and compared to the species, the depth is different levels of species specializations are closely related to the environment conditions. Chetodon is the major fish on coral. It can be used as a guide to assess and monitor condition of coral reef. The next is Acanthuridae, found around coral reef, over 10 bottom, shallow, and near the surface. The highest species diversity and are often abundant in coral reef areas. So let me just maybe you can consider So my conclusion is the percentage of specialization between coral and non-coral will differ in other locations. The highest is coral with alga, followed by coral, and then total of 670 individuals, of which from 17 species and 17 families. Species that can be found around types is Gedung, Lantai, and atau. Lantai is a family hybrid variety with total 25 species and 161 individuals. Fisheries observed the highest number of individuals, which is 360 individuals. Fish were associated with struts at each side with relevant background, included as a surface on which specific food was stored and served. So the design of this store and other sites also vary. Mrs. Williams. And everyone. Tetap dulu. Bisa begitu point shop. Okay. Yeah. Hello everyone. My name is Udayana. I'd like to talk about exploration of associated biomass of the Untuk in the coral and farming facilities of City Bali Aquarium. So the Untuk is a member of family Zoanthidae class of Anthozoa. Next. Okay, so this research is based on the increasing demand of marine ornamental aquarium. So people pay high amount of money in order to get most beautiful coral and invertebrate species. Thus, it generates $1 million annually. And okay, so yeah, so trade of life marine organism promotes the movement of these organisms worldwide, and it has been considered that it can have potential to be pest of the main organism in the aquarium or becoming invasive species. Next, please. Okay, so this is the Untuk. So the Untuk is one of the most popular taxa for marine ornamental aquarium. Ecologically, it plays an important role in marine organisms, such as providing shelter for other invertebrates. Next, please. Okay, so this research aims to examine the associated biota and their potential established relationships with the Untuk. Next, please. Okay, so we sampled 35 of colonies of the Untuk, which have been culturing in land farming facilities of City Bali Aquarium in Denpasar. So the term of associated biota in this research refers to the organism living attached to the colony or hiding between the tentacle of the colony. So for the attached biota, we firstly we transport the colony into the smaller aquarium and then we spray with water. Once they're not so attached to the colonies, and then we pick with the tweezers and then we preserve. For the hiding biota, we also transfer the colonies into a small aquarium, and then we generally shaking the colonies, and then once they came out from the colonies, and then we pick with the tweezer, and then we preserve. And all the identification are based on morphologically. Next, please. And following the identification, we found there are 4 groups of invertebrates. They are crustacean, polychaete, flatworm, and cyclogonids. Next, please. And okay. Okay, delay.
Yeah, so these are the specimen picture after we research. Then next, please. And we classify the associated biota according to their feed habits. So we assume that all the crustacean and polychaetes are omnivorous. In contrast, the pycnogonids and platform are parasitic. Next, please. Regarding the feed habits, the Untuk itself is a heterotroph, so it is a plantivore and has symbiote at the same time. Next, please. So, according to those feeding habits, we construct the potential role and relationships into Untuk's associated biota. They are consist of 4 relationships. First is shelter protection, second nutrient transfer, third nutrient cycling, and fourth is food source. For example, the potential relationship between marine spider, or the group of pycnogonids and the zooanthus. So zooanthus serve as a shelter and also as a food source because the pycnogonids eat the polyps. Conversely, the pycnogonids, when they are hiding among the colony, among the tentacle, and also crawling above the colony, its metabolic waste can be the nutrient cycling, the nutrient source for the alga and the symbiont inside the zooanthus. Okay, to conclude, so far the associated biotas of the zooanthus consist of crustacean, polychaetes, platform, and pycnogonids. And the second, the relationships between the zooanthus and the associated biotas are potentially mutual, commensalism, and parasitic. Next, please. Okay, so we are very thankful for City Bali Aquarium for all the support and facilities. Thank you very much.
Next, we will to Mr. Agustinus Widodo. Please.
Bapak pakai pointer ya? Terima kasih Bapak Ibu, senang bertemu dan berdiri di sini. Thank you very much, Chair. Very glad to be here because I think I always here. Nama saya Agustinus Widodo. I think this research done by more than 10 media groups, and one of author here is Ibu Hetty. So please, Bu Hetty, help me if there is a question that's very not very okay. Thank you very much. My research title is estimation of number and position of fish aggregating devices in Indonesia accumulating water. Next one. Okay, then we know that I think most of fishers in Indonesia now, especially for the inline, full inline, full sand, and also some fisheries using fat for their fishing. And then unfortunately the idea due to many of fact. So government of Indonesia here is KKP. I think raised the sum of degree since 1997, I think, and then the most recent degree on the fact in 2020. But however, despite this regulation, adequate implementation and enforcement have not been achieved. So that's why we try to research to get information number and the position of the fish, because there are no information about that for the management purpose. This is the figure one is present the fishery that are using PET in Indonesia, especially in Indonesian archipelagic water. I think I'm not sure that you know or not the Indonesian archipelagic water in Pacific Ocean, Indonesian Pacific Ocean. Kita menyebutnya WPP 713, 714 Indonesian Fisheries Management 730, 714 and 750. This is the figure of the fishery that using practice in the fishing practice that like hand line. I think you know hand line, like that. And then troll line, kita bilang tonda, and then of course pull end line. I think we call tuhati. I'm not sure you know or not. And pulse in Bali is made of pulse. Next one. Okay. This is the archipelago consists of Indonesian fisheries management 7 here, 7 in Banda Island, Banda Island, and then Banda Sea, and then 175 in close to the Manado. Okay. I think the FED, is handmade of party attractor designed to lure or attract and facilitate their aggregation around the object. This is I think a figure of FED here. FED is fish aggregating devices. Yeah. We use the anchor FED that consists of a buoy, and then like line, very long line, a long rope, sometimes until 5,000 meters, I think, more than yeah. So and this is anchor and weight. And to anticipate the risk of damage due to the current and oceanographic conditions, or fishers mostly they deploy the FED with 1.2 until 1 times of the sea depth their mooring, their line. So very long. That's the consequences of it in practice. The buoy will move about 250 meters round, something like this. So this is I think the situation in the sea. Okay, the data. We are very tends to get data from the fishers, so we trying to get data from the satellite. I think this will a long explanation about this method. So I think we just want to say that this is the area that we collect data, the imagery data. Okay. Okay. This is analysis of our methods. I think this is I think skip because not really important, but I think it's very quite long to explain. But if you want to know, I think you can ask later. Okay, next. Okay, this is also still our methods. We doing we done the data collection into time, June in as T1 and August in T2. Okay, next one. Okay, this is I think result in discussion. So the number of paid in FMK 713 was 661, and in 714 was 608, and in FMK 715 was 291. This is the estimation of the distribution and the number of the package. This is 713. Mostly they deploy in close to the Sulawesi Tenggara. Okay, Sulawesi, and then even for AFM 012, I think maybe the state in all of then yes, okay. I think it is within this. Okay, okay. I think this is distribution in 715. Okay. Then this one. Okay. The mostly of this one is between states, mostly under very close, under maybe under type nautical. That's I think not good, because our degree should be 10 nautical. This is very between distribution between set in 714. Okay, next this is in okay. Our conclusion. This one in the research I estimate that there are about 661, 608, and 291 units of it in FMR 713, 713, 714, 715 respectively, and mostly of paper between mostly under 5 nautical means. That's we would like to acknowledge from this. I think that's our contribution. The acknowledgement. This research activity conducted in 2021, and I think it's performed by funding by MDP Foundation. Thank you very much.
Okay, we will next continue to the next presenter. Presenter in this presentation. Okay, to Mrs. Happy Hatati.
Thank you very much, everyone. Thank you for coming to our presentation today. Our research is historical and documenting unlimited online teaching in the Pacific Ocean in Southern Udayana. That's for unique award online teaching. And my name is Kedra from Udayana. As an introduction, Udayana or Clinical Basis is one of the main target species for Indonesian phenomenon fisheries, mostly in Eastern Indian Ocean. And the commercial phenomenon fishery has begun in 1960, and the deep water manufacturing was introduced in 1980. And Udayana started as a target since the techniques introduced. And however, we also know about its abundance and size distribution, especially in the northeastern Indian Ocean, which are top fishing grounds for Indonesian phenomenon. And its features provide updated information and nominal frequency and size distribution in the Eastern Indian Ocean based on Indonesian phenomenon. For material and method, we use imitation scientific observer data from commercial tsunami vessel based in Benua, Philippines. And this program started in 2005 through an Australian collaboration project. And since 2010, it has been conducted by the Research Institute for Universities. And the data was collected from October 2005 to December 2021. But the data collected in 2005 were excluded from this analysis because it was at the beginning of the trial of the program. And the size measurement was taken in Falklands. These data were plotted according to longitude, latitude, altitude number, and length. And the length frequency was aggregated and average length was analyzed throughout the period of the position. And the nominal CPUE was calculated as the number of crystals per 100 groups. And the data were plotted on 5 by 5 degrees per day. As for the results, from the data set, it contained 118 trips, 3,180 steps, more than 4 million hooks deployed, and hooks between both range between 10 to 15 during 2006 and 2021 observer trips. And from the figure 1, we can see that from 2006 to 2013, the CPUE was relatively stable on 0.21 on average, and the highest, and it increased to the highest CPUE in 2014, which was 0.29, and it dropped into 2021 to nearly 0.09 feet per 100 foot. So the highest into the high CPUE of the Udayana in 2014, as you can see in the table 1, is probably caused due to the vessel on the script on that year is targeted Udayana on their assistant script, because you can see that it has the highest hooks between flows, which is 50 in the time of period of occupation. And also you can see here in the special degree of Udayana, the effort was distributed between 0 to 35 degrees south and 75 to 120 degrees east. And you can see in the 2014 that the fishing area is focusing in the higher latitude, while other trips in other year is mainly further south. And for the size distribution, in total, we measured about 8,204 feet. And the mean length of the Udayana coast is 170 centimeters of length. And overall, the average length of Udayana from 2006 to 2021 is increasing, precisely increasing. And this is a special distribution of Udayana to see the composition of the by the level of the maturity. So as you can see, the authenticity for Udayana is 100 centimeters of length, and the picture show that the composition of between the mature and immature fish is more balanced west further west than the coastline. So bigger fish thought mostly is near the coast. And for the conclusion, the trend of CPUE of Udayana from 2006 and 2021 was declining, and the average size of Udayana was decreasing. And this is very interesting finding because it might caused by many factors, especially from biology parameters. We can explore more or study more for the future work. And we acknowledge the ITF, the Institute for Fisheries, the entire HR and FFA for funding and for this research. Thank you for your time and attention.
Okay, all the presenters stand for your research. For the next, we continue to the discussion session. We have 8 participants for the discussion session. Or maybe you can raise your hand, okay, and stand up. Mention your name, institution, and mention who your question is for. Okay, in our first session, I will open to 3. Question.
Good morning. My name is Manuela Pello, and I work in IRB side. And also I'm here because I feel the housing. And so I have some questions for different folks. So if I can ask multiple questions, is it allowed? Yes. So I have a question of first presenter. Yes. So it's about the business, the method that you use to handle it. Do you think that the selectivity of the year has affected your results? And for example, there are some countries that avoid business and so. And what do you expect about the impact of this selectivity on your results?
Is it clear? For a question? You can no. What is that?
Maybe again,
Okay.
pertanyaan
Second
ya.
question.
So the second question is the so it's
Tadi buka.
Yeah.
So you come to business, and my question is whether you expect to identify more issues when your sample size is larger. What do you expect the impact of the sample size with respect to the number of size? Then I have a question for the board.
Yes.
So, your study was in a trial, yes. So I was wondering, in natural selection, would you expect similar results? Or and last question.
Thank you, Manuela.
It's about the ground truthing. Ground truthing your results. So demonstrate that your findings are real. Maybe how can you test the validity of your approach? Maybe it is possible to conduct some interviews with the teacher. Okay. To validate. Yeah. Thank you.
Okay, thank you to Mrs. Pamela Costello. Maybe for the presenters, you can answer the question. For the first is from oh, yes, you can answer.
think there is of external that we would to content in the case. is quite enough because we use varying sizes, right? From half until 3 centimeters. So varying sizes. Yeah. So the fishes, the size of the fishes is different, right? So I think it can it can catch all the fish. And we put the experimental unit at 5 p.m. until 7 p.m. So it hope we hope that diurnal fish and nocturnal fish can catch the second question for Gedung.
Yeah, thank you for your question. So yeah, I also expect that by adding some samples to our data, so I can get maybe new species, but it is hard actually to catch the fish underwater. So when we conduct the research, I also have a proposal how to the fish. And honestly, I also didn't expect that I will get a new sequence for that fish because the Gilman Bay is really near the big port on the Gilmano, and a very intense activity of the fishery in there. So that's really beyond my imagination that I will get a new sequence that we can really add it in the sequence. So yeah, I think we need to doing more research for that. Yeah, I already thought that a low economic value coral reef trees that maybe some researchers not really um, uh, really want to study about that fish. Yeah. Yeah. Yeah, but some of the aquamarine fish like Bombay carcinopsis, that one is marine ornamental fish and have a high price on the market. No. Yeah, but only the Bombay carcinopsis because this fish is endemic from the Bombay archipelago, and fish because this fish has a high price, and now the government and Fisher already introduced this species in other areas. And the Gedung is one of them.
Okay, thank you, Mr. Gedung, for the presentation for thank you for the Okay, thank you for your mind-blowing question. So it would be new idea for the next lesson. Okay, so I think those organisms that we found in as a relationship with the answers, I think they have been adapting to the aquarium conditions. So I think the result will be different with those living in the natural environment. Yeah. So thank you for the new idea. Thank you. Okay, thank you, Mr. Jessica. Next, last question is for Mr. Aditya.
Terima kasih, Bu Manila. Thank you very much. Thanks to share. Very good sharing this meeting. Yeah, Iguanella is actually the validation already using the ground team, but I didn't inform in this method. But we also using the like apa, pesawat yang nggak pakai awak itu. We zero from the our research people, and then we trying to get photographs in position something like. So I think you can ask to Ignatius because I think he collect the some of the data maybe still collect, and if no I think we can discuss later on because yeah I think and thank you very much but maybe very limited site that we collect the data of validation with validate this but we already trying to get a validation from the okay thank you very much.
Thank you very much for the because our time is over. The online there are okay, I will ask them. Okay, first, Mr. Arif Taslihan. Can you hear me?
Yes.
Okay. For the first presenter, maybe you can present your thank you, Sir Arif.
Yeah.
Yeah. You can start your presentation.
Okay. Thank you. Good morning, everybody. So my presentation is on Dahlia tuber from Dahlia variabilis as a source of inulin for the control of acute hepatopancreatic necrosis disease in shrimp. So this is the second year of my project. For the first year, we extract from the Dahlia tuber, and now we apply to control the disease. Why we use inulin? Because inulin is what you call like prebiotics. So prebiotic is defined as substance that cannot digest by host shrimp, but it can be digested by some microbia in the digestive system of the shrimp. My background is so my background. Next, please. Yeah, so the acute hepatopancreatic necrosis virus is one of the most dangerous diseases. So frightening the farmers that culture shrimp because it causes mortality during the first month. Maybe from the 2 weeks after stock, so can get mass mortality because of this disease. So why we use inulin? Because inulin is a prebiotic. It can be digested by bacteria in the tractus digestivus or digestive intestine in shrimp. So the bacteria in the intestine will vary, not only dominated by Vibrio. And as you know that Vibrio cannot use inulin. Yep. But some bacteria, useful bacteria, like lacto acid bacteria, they can use inulin. So they, we hope that this lactic acid bacteria can dominant in the intestine of the shrimp. So in the inulin has been widely used in relation for food ingredient for human health. So it can prevent like cancer, diarrhea, and so on. So during the first year, we get the inulin using the methods we call as gravitation, cold gravitation. We get almost 8% from the tuber of the dahlia. Next, please. The method to conduct the, this is the inulin, so this is the tuber. So we blend first and then heat for 80 degrees Celsius, and then filter. Then we filter, we collect and keep in the refrigerator for almost until 3 days. So we get the deposit in the bottom, whitish. So it calls as inulin, raw inulin, and we dry it, and we can use this inulin. Next, please. So the methodology is we do the experiments. So we use tank. Capacity is 700 liters, filled with seawater, sterilized with the chlorine, and then stopped with 100 PLs of Vaname, PL30. Then we treat the shrimp with feed that contain inulin extract from dahlia tubers as 2.5% and 5.0%, and then we compare to 5% commercial inulin, and we compare with no inulin as a control. So its treatment given 2 replication. So the feedings of inulin last up to weeks, and then we continue reading until 45 days. And then at the end of the study, we take 5 prawns and dissect, and there is the scene where homogenize and implant in MRS media to see lactoacid bacteria, and also implant in TCPS agar to see the vibrio. Then after each treatment, shrimp were challenged with Vibrio parahemorrhagicus, the strain happen AHPNT that cause the disease at density of 6.6 FU per milliliter, and the shrimp mortality was observed for 96 hours. And the live shrimp will then proceed for histopathology to feed the microanatomical structure of the gut and also hepatopancreas. Next, please. So this is the result of the experiment. So there is no significant difference about the survival rate, final weight, length, and also weight gain of the all treatment. Yeah, next, please. So in the TCPS agar, the left picture, so we can see that in control, that zero inulin, they dominated by greenish colony. But for the inulin treatment, there is dominated by yellowish colonies of vibrio. And the right pictures, we see the lactoacid bacteria grown on the MRS media. Next, please. So the result of the growth in the lactic acid bacteria and vibrio. So in the treatment of INU1, INU2, INU3, INU5. So we can cut 1.4 at the control of zero inulin compared to the 2.1 and 2.3 in inulin treatment, and also 5.4 CFU times 10 to the 5 CFU per milliliters. And total acid bacteria or LIP, there is 0.6 the low content of LIP in the control or zero inulin, and on the 2.5% inulin in the feed, so we get 3.2 LIP times 10 to the 2 CFU per milliliter, and for the 5% inulin we got 0.1 LIP, and the highest is treatment with 5% commercial inulin in the MRS and LIP, 5.2 times 10.2 CFU per milliliter. So next, please. So for the challenge test, so we got the interesting is in group INU1 or zero inulin. So the mortality of shrimp observed after 24 hours post challenge with Vibrio parahemolithicus strain happen, and at 48 hours post challenge we got 80% mortality, and 72 hours we got 100% mortality. While for the treatment with INU2, INU3, and INU4, there is no mortality observed in the treatment. So next, please.
Okay, sorry, Mr. Arif, your time is over. Maybe you can conclude your.
This is the last, yeah. So the conclusion is inulin is very effective to control happen during this my experiment. So there is further experiment in mass scale, yeah, for this using of inulin to control the happen. So thank you very much.
Okay, thank you very much, Mr. Arif. Okay, we will go to the next presenter. To Mrs. Lantai Tampanalo. Can you hear me? Mrs. Bunga? Mrs. Bunga, can you hear me?
Sorry. Okay, can you hear me?
Okay.
Yes.
Good. You can start your presentation.
Thank you, moderator. Here I will try to present the result of our research with title Effect of Alginate Concentration on Viability of Probiotic in Symbiotic Microcapsule Beds After Incubation at 70 degrees Celsius. I'm Bunga Rante, and this our team. Next slide, please. The introduction. The use of probiotics in various sectors has become common, as well as the use of probiotic in aquaculture. In addition to improving the water quality of cultivation media, probiotics are also expected to improve growth performance. This is possible because some probiotics isolate can produce enzymes that can help cultivant absorb the food given, improve the intestinal bacterial community, suppress pathogenic bacteria growth, and increase cultivant immunity. To achieve this goal, the probiotic application method in aquaculture is needed. Mixing probiotic in feed has been done by many cultivators, but the mixing is still done manually or after the feed is molded into pellets. To certainly inefficient
because many things can be source of contaminants and damage the viability of prebiotic. On the other hand, adding probiotic or symbiotic to the feed formulation before pelleting will be constrained by the high temperature stress of the pellet molding machine. Microencapsulation is one method of presenting probiotics and prebiotics into a symbiotic product that can overcome environmental stress problem such as temperature or contaminant. One microcapsule method widely used in food sector is the encapsulation of probiotic in alginate bits. The method technique is through the extrusion method. Based on the description above, this study aims to determine the viability of probiotics in symbiotic microcapsule bits using several levels of alginate concentration after exposure to a temperature of 70 degrees Celsius for 60 minutes. This expected to be information for mixing symbiotic in feed formulation before pelletizing. Next slide, please. Next. Material and methods. This research start at rejuvenation of probiotic isolate. This research are conducted at the Fish and Environmental Health Laboratory, Fisheries Research Center for Brackish Water Aquaculture and Fisheries Extension in Maros. The study began with rejuvenating probiotic Bacillus subtilis in TSA plate media, then continued with the multiplication of probiotic in liquid media nutrient broth. After incubating the probiotic on the incubator saying for 24 hours, the probiotic cells were harvested by centrifugation at 6,000 RPM for 10 minutes. The precipitate form is utilized with physiological solution one time. After rinsing, the probiotic cell precipitate into 1% inulin solution. This symbiotic solution is then incubated for 20 minutes and on top of sewing incubator. After incubation, the symbiotic solution is transferred into the air layer, which contain the alginate solution according to the treatment, 1%, 2%, and 3%. Its symbiotic suspension was homogenized using a plate stirrer for 10 minutes, then printing using a 20 gauge needle and drop into calcium chloride solution. The microcapsule bits format are then filtered and stored in its air layer according to the treatment. Next. Miss slide maybe. This study design use a complete randomized design using 4 treatment and 3 repeat. Control, we use probiotic without encapsulation or free cell bacillus. Treatment A is symbiotic coated with 1% alginate. Treatment B is symbiotic coated with 2% alginate, and treatment C, symbiotic coated with 3% alginate. All of the polymer with this microcapsule symbiotic bits, we incubate in incubator water bath at 17 degrees Celsius for 16 minutes. Then we observed modifier at the shape and diameter of symbiotic microcapsule bit and density of probiotic in nutrient bit, symbiotic microcapsule bits before and after export to temperature at 70 degrees Celsius for 60 minutes. The data were analyzed with SPSS application. The result of our study showed that The morphology of microcapsule symbiotic beads was round to oval, transparent white in color, with a degree of suppleness and the concentration of alginate added. The diameter of symbiotic granule is 900 to 4,000 micrometer. It still include in the microcapsule category based on the classification of microcapsule diameter by Lachman and Nevedic. The result of observation of the probiotic population and symbiotic microcapsule bead range from 1.23 times 10 to 7 CFU per milliliter until 1.11 95, then 7 to 5 to 7, 10 to 7 CFU per milli. The difference in alginate concentrated had no significant effect on the probiotic population and symbiotic microcapsule beads. The result were obtained after the symbiotic microcapsule beads were incubated in the water bath at 70 degrees Celsius for 60 minutes are shown in this figure. Based on the data in this figure, it can be seen that there is a decrease in probiotic population after incubating at temperature 70 degrees Celsius. The highest population decrease was found in the control treatment, namely probiotic suspension cells that were not coated or flea cell, which decreased by 32.35%, followed by treatment D decreases by 27.49%, and treatment F fell by 25.93%, and the lowest in treatment B only decreased by 18.05%. The variety analysis results showed that the alginate concentration significantly affected by viability of Bacillus subtilis in symbiotic macrocapsule beads after incubating at 70 degrees Celsius for 60 minutes. The result of the follow-up trial showed that the decrease in probiotic viability in 1%, 2%, and 3% alginate concentration treatment was not significantly different, but significantly different from the control treatment without encapsulation. Next slide. The conclusion of our study, based on the research, it can be concluded that alginate concentration of up to 3% can suppress the rate of decline in population of Bacillus subtilis after exposure to a temperature of 70 degrees Celsius for 60 minutes, so that it can be used as a reference as an effort to present probiotics that are safe and easy to apply. This data also allowing testing even higher temperature stress to obtain maximum temperature stress value against Bacillus subtilis in alginate macrocapsule beads. Thank you.
Thank you very much, Mrs. Marante. Okay, we will continue to the next presenter, Mrs. Enda. Can you hear me, Mrs. Enda?
Yes, I am here.
Okay.
Sorry, I off my camera because my internet unstable with I on my camera.
Okay. Okay, you can start your presentation.
Okay, thank you. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh and good afternoon. Thank you for moderator for the chance to given for me to present our research in this this time we will discuss about application of wholesale microalgae as an alternative to prevent fibrosis. My name is Endang Susianingsi, and besides me, there are Mrs. Inci Ayu Khairana Kadria and Mrs. Nur Hidayah. We, as a team in our research project, we are from National Research and Innovation Agency. Okay, we start the discussion from introduction. As we know, the classic problems that cause the high mortality and low shrimp culture production are shrimp disease. There are so many shrimp diseases like parasite disease, bacterial disease, and viral disease. The disease occur not only in hatchery, but they can occur in grow-out ponds from extensive to super intensive culture system. One of the disease is fibrosis. Fibrosis is a bacterial disease caused by Vibrio species bacteria. The disease can cause a decreased growth, a high mortality, and so they can decrease shrimp quality. Until now, the prevention of major disease caused by pathogenic bacteria has been carried out by applying antibiotic. But if we use antibiotic, they can causing a new problem, because they can increase of resistance bacteria in environment. So, based on this, we try to find some method, especially safely method to prevention this disease. One of the method we try to use natural antimicrobial. Slide please. Okay, research on the use of natural antimicrobial agents for the prevention of fibrosis has been widely conducted, and so that the use of natural antimicrobial agents has the potential as anti-fibrosis. One of them is by using microalgae that have the ability to produce bioactive compounds that have antimicrobial activity against Vibrio bacteria. So this is research. This aim of this research was to determine the ability of microalgae as an alternative anti-fibrosis for disease prevention, especially in shrimp culture. Slides, please. Next, please. Okay, to research the aim, this is slide about the material and method used in this research. The ability of microalgae as antifibrosis in this research was carried out through a challenge test. We using pathogenic Vibrio bacteria. We divide this research in two groups. The one part in vitro test. In vitro test, we use 5 types of microalgae test of 5 type microalgae, namely Melosira sp, Phaidodactylum sp, Nanochloropsis sp, Porphyridium aerugineum, and Porphyridium sp. We use Erlenmeyer and we fill with sterile seawater with a salinity of 20 activity as much as 100 milliliter. After that, we given microalgae with a density of 10 to 5 cells per milliliter. Challenge test, we using fluorescence pathogenic Vibrio bacteria with a density of 10 to 7 CFU per milli. This research, we use a complete random design with 6 treatment. 5 we use 5 types microalgae, and 1 treatment we use as a control, and we repeated 3 times. Okay, the second part is in vivo test. The same with the in vitro test. In vivo test, we use the same microalgae. We use 5 types microalgae, and we add with two control. one control positive and one control negative. From as animal test, we using tiger shrimp with density 20 individual per storage, and we filled with sterile seawater at much 2 liter and salinity 20 activity. We infected with Vibrio pathogen at a density of 10 to 7 CFU per milli. The parameters observed include the growth of Florence pathogenic bacteria on the test media every 24 hours for 120 hours, and the survival rate of shrimp in the in vivo test. To know the growth of the bacteria, we collect a sample from water and we culture on TCBSA media by stratified dilution. And after 24 hours, the growing bacterial colonies were counted using the Buller method. The data obtained are analyzed statistically using graphic observation and calculation of larva survival inside the maintenance container every 24 hours. Okay. This is slides about the results of this research. In vitro test, we can see in this graph, it can be seen that the several types of microalgae given in whole cell have the ability to inhibit the growth of pathogenic vibriobacteria. In treatment with Porphyridium aeruginium, pathogenic vibriobacteria were not detected at 72 hours after infection. The presence of pathogenic hours after infection, the presence of pathogenic vibriobacteria was also not detected in treatment with Melosira sp at the 96 hours after infection, and Nanochloropsis sp at the 120 hours or at the end of the study. The concentration of pathogenic vibriobacteria at the end of the study was 10 to 3 CFU per milliliter. Next, please. Okay. In vivo test, we found, and we can see in this graph, this is graphic about the growth of pathogenic vibrobacteria in the challenge stage, in the challenge test using whole cell microalgae and tiger shrimp. In this, from this graph, we can see that administration of microalgae Porphyridium aeruginium and Phaidodactylum sp is able to inhibit the growth of pathogenic vibriobacteria better than other types of microalgae and control. It can be seen that the density of pathogenic vibriobacteria at the end of the study or day 5 in treatment with Porphyridium aeruginium and Phaidodactylum sp microalgae was 10 to 2 CFU per milli, while in the control treatment, the density of pathogenic vibriobacteria was 10 to 3 CFU per milli. This indicates that the two types of microalgae are able to inhibit the growth of pathogenic vibriobacteria. Next, please. This is graph about the percentage of the survival rate of the shrimp in the challenge test using the whole cell of microalgae. From this graph, this graphic, we can see that the whole cell microalgae administration has no effect on the percentage of survival rate of test animals. This can be seen at the end of the study where the percentage of survival rate in the treatment with microalgae administration. Okay, sorry. Yes. Okay, we jump to conclusion. Sorry, the slides about conclusion. Okay, wholesale administration of microalgae types, Porphyridium aeruginium sp, Melosira sp, and Nanochlorophysis sp can inhibit the growth of fluorescence pathogenic Vibrio bacteria in in vitro tests. And in vivo tests, we found that wholesale administration of microalgae Porphyridium aeruginium and Pyodactylum is able to inhibit the growth of pathogenic Vibrio bacteria better than other types of microalgae and control. Okay, that's all my presentation. Thank you. Sorry for taking your time. Okay, thank you.
Thank you very much, Mrs. Enda. We will continue to the last presentation. Is Mr. Ibnu here? Can you hear me, Mr. Ibnu?
Yeah.
Yeah. Okay, now is your.
Great. Thank you very much for the times. So it's my voice is okay?
Yes.
Okay, thank you very much. So today I would like to talk about feeding management and recirculating aquaculture system for spiny lobster. Okay, the next. So there is the most important area when we want to do nursery for lobsters. The first one is the environments, how to make good environments. So here we propose to use a kind of recirculating systems. We call this multi-loop filtration systems. The second one is feeding managements, because when we get a good feed, formulated diet, the next one is how to manage the feed appropriately. So here, we want to know how to adapt the formulated diets for baby lobsters in the appropriate way. Okay, the next one. Next. So this is our prototypes of recirculating system. We use around 25 and 30 black tanks, and then this is the pictures of our prototype of filters. Next. Just one click. Okay, this is main loop of the filters. The main loops is the water come from the reservoir and come to the ring tanks and back to reservoir. The second one, second loop is water from the reservoir come to the filters, several types of filter, then come to the reservoir. The third loops is the water come to the third chamber and come to mechanical filter and come to the reservoir. Okay, the next one. So here we want to see the water quality inside inside the reservoir. This come from the reading tanks. The next one is the water come from mechanical filter and phytobiological filter, and then the biological filter, and the last one is come from a mechanical filter. Okay, next. This is the visualization of the second loop of the RAS. We make it because it's more complicated than the other ones. So here the water comes from the reservoir and attacked to the polyester fibers by perforated pipe wrapped by green screen, and then this come to Caulerpa lentilifera. Here we put to become biofilters, and then from here the water come to skimmer, and then from skimmer come to biological filters. This two box is biological filter made by gravel and coral stones. This is not based on model scales. So if we want to see the real one, we can send you the pictures. Okay, next. So the experimental designs, actually we want to know how to adapt the formulated diet to lobster, because the lobster cannot eat direct formulated diet when it's still in its baby stage. So here we want to check the survival rates and growth rate from these different feeding management. The first one is formative diet, and then the second one, an increase 10% every day, increase 10% every 2 days, increase 10% every 3 days, and the fifth one is fresh diet only. We get this porelli or baby lobster is from Pulau Raya in Aceh, around 0.31 grams, and then put in 25 of the tanks. Okay, next one. Okay, this is the procedures. You can see, if you want to see more, you can see my PPT. Okay, next. So the observation variables is the first is mortality and the molting. Every day we count it every day, and then second one we weight it initial day, weekly, and the final day. We also measure the water temperatures and another parameter of water quality, and then the fifth one we check the growth of biofilters of Cholera lentilifera. And then we'll calculate the survival rate, weight gain, and specific growth rate. Okay, next. We do the statistical analysis by SPS versi 20, and then do Duncan post-hoc test after 1 week ANOVA. Okay, so then next. So the feeding response for porelli is quite good. When we put the formatted feed directly to the water, they can catch on less than a half minutes. Okay, then next. So based on statistical analysis, we see here that the D3 or adaptation every 3 days, 10% increase the fresh diet every 3 days is the best specific growth rate and also weight gain, but not for survival rates. Compared to, for example, the fresh diet is lower, the lowest specific growth rate compared to adaptation diets. Okay, then. And therefore, here we see that lobster decreases survivals mostly in the porelli stage. This the first week, this porelli stage transfer to the post-porelli stage only in a week, and the mortality increase sharply. And then after the first week, the mortality is not increase sharply. It's quite stable. Okay, next. Okay, here the first day and the first week, we can see that the cumulative molting is quite high compared to the other ones. After first weeks, the molting is quite stable. Okay. Next one. So we found that after the research, termination of the research, we see that ornatus is quite more Paludis ornatus, mutiara or pearl spiny lobster compared to bamboo. Opposite to Lombok, when in Lombok it's many homarus than ornatus. Next. Next. So based on general water quality profiles here, this is quite good. Next. So when we compare several variables for total ammonia nitrogens, nitrate, nitrate, and dissolved oxygens, pH, and temperatures, the total ammonia nitrogen for fetal biological filter is quite lower than the other ones. Also, we can see here the tendency for nitrate and nitrate is also lower, but for the dissolved oxygen, because in the night they consume the oxygen, is dissolved oxygen is decreased. The pH increasing because they take the carbon dioxide from the water, so the pH increase. So the temperature is higher because the circulation in the phytobiological filter is not quite often changed. Next. Here, based on the biomass, the biomass increase the first week is very sharply. very sharply, more than 100% in a week, but decrease for the second week until the end of the research. Is because of the feed consumption is decreased. It is because of mortality is increased in the first week. Okay, we can see here this is the first week from 2 phone until 5 phone. We can it can increase from 2 phone until 5 phone of Caulerpa lentilifera in a week. This is the end of the population of Caulerpa. We get 600 grams of Caulerpa in 1 month. Okay, next. So we conclude here that based on the adaptations, I recommend that if we want to give the formulated diet, we can adapt every 3 days by 10%. To increase 10% of formulated diet and decrease 10% of fresh diets. And we need to take more attentions in the first week or in the stage between porous to post-porous stage. And then we need to confirm more whether Panurus ornatus is have more population than Homarus in Aceh compared to Lombok. And the last one, we can increase to use the Caulerpa lentilifera in the systems. So we recommend to make like us use a multi-loop system like aquaponics. So we can see we can this we can call this seawater aquaponics. Okay, next. So we want to thanks to ASIAR, Australian Center for International Agricultural Research, for funding, and then also for Gondol Mariculture Research Institute providing the diets from this research. Thank you very much.
Okay, thank you very much, Mr. Untuk, for the presentation. Okay, for the next, we will open the discussion session. We have 10 minutes. The discussion. Actually, we have 3 questions. Here or in my session, you have any questions? In this Zoom room, do you have any questions? No. This room, do you have any questions? No. I think all is hungry. I have a question for Mr. Enda. Can you hear me? Mr. Enda?
Yes, here. Yes,
Enda?
I'm here. Yes.
Yes. In your research, you use TCBS media.
Apa? Yes.
Yes. Okay. I want to ask you about what's the most important thing to culture bacteria by TCBS media to avoid contamination?
Apa? Can you repeat, please? Because it's not clear. The sound is not clear.
What's the most important thing to culture bacteria by TCBS media to avoid contamination?
Oh,
Can you answer?
I think TCBS media is already a specific medium for Vibrio. So another species beside Vibrio cannot grow on TCBS medium. So it's because this is a medium is very specific only for Vibrio species. Answer your question. This is my answer is answer your question.
Okay, thank you, Mrs. Enda. I think enough. Yeah, the time is over. Okay, my honorable Mrs. Enda and in this room and live. We finally come to the end of our session. On behalf of the committee, I would like to thank the presenters for the insight and input from our participants during this session. For that, please give a round of applause. Thank you for the good work, and I will see you in the next event. Stay home, santai om. Wassalamualaikum warahmatullahi wabarakatuh. Thank you.
Thank you very much.
Next slide is floor.
Oh, kembali ke lantai 3, Mbak.
Okay, thank you.
Maaf, Bu, nanti saya nggak bisa dengar.
Ya,
Recording in progress. Recording stopped.
Will begin after 4 to 5 presentations. The allocation time for this presentation is 8 to 10 minutes. Without further ado, we are going to start the presentation now. Please welcome our first presenter for Mr. Manja Mekibon to deliver his presentation. Okay, for Mr. Manja Mekibon, time is yours.
Okay. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody, ladies and gentlemen. Is that my voice is clear enough?
Yeah, clear.
Okay, thank you. I would like to presenting my study about the determination of withdrawal time, efficacy, and safety tests of enrofloxacin in goldfish. Next. For introduction, we have a goldfish because goldfish is consumes in fresh water, which is kept since 475 BC in China. But in Indonesia, it has been kept in 1920s. The origin is from China, Europe, Taiwan, and Japan. So that there is now 10 types of carp in Indonesia based on morphological characteristic is available in Indonesia. So the fish farmer for problems in the field was fish disease caused by pathogenic bacteria. One of the pathogenic bacteria is called red sore disease by Aeromonas hydropila. So this group of motile Aeromonas septicemia can cause up to 80 mortality in short period, about 1 to 2 weeks. Okay, And fish farmers usually use antibiotic for prophylaxis bacterial infection. For example, the enrofloxacin, group of fluoroquinolone, actually is first used for veterinary medicine, which is the main metabolite is ciprofloxacin. Next. So if we excessive and continuous use of enrofloxacin cause antibiotic residue in fisheries product and bacterial resistance to antibiotics. For example, prophylaxis fish with enrofloxacin so need to be controlled properly. That medicine function effectively and would not cause antibiotic residue above the maximum residue limits. Because some of importing country still tolerate certain types of this antibacteria with reference to the quality standard MRL. So we need to determination of withdrawal time must be carried out systematically and complete with scientific principles to gain food safety requirement. So the study aims to determine the duration of administration, efficacy, and safety of enrofloxacin in goldfish. Next. This is some materials and equipment that our study continues. Next. So the design of this method for the efficacy and safety test, we divide into 4 groups: the negative control group, positive control group, efficacy test group, and safety test group. The negative control group, the goldfish are kept in running media without being treated with bacteria, being injected with bacteria, or without being given antibiotics in pellets. But in the positive group, the goldfish are being treated with bacteria, with Aeromonas hydropela isolate around 10 CFU per milliliter per fish. We injected isolate of Aeromonas hydropela. And for the efficacy, we use injected bacteria Aeromonas hydropela and given treatment with the recommended dosage and method. The recommended dosage is 1 gram antibiotic per 100 gram of pellet or fish feed. And for the safety test group, the goldfish are kept in rearing medium, and feed is given 2 times from the recommended dose or 2 times from the efficacy test group. So we have 30 fish per tank. We rear in 2 meter cubic fiber tank, and we acclimatize for 3 days, feeding 3 times per day. Next. The withdrawal time test, we have divide 2 group, control group and treatment group. About 150 fish per tank. We rear in 2 meter cubic fiber tank, feeding 2 times per day for control group with ordinary feed or pellet. For the treatment group, we added or mixed the feed with enrofloxacin by 1 gram per 100 gram. We feed 2 times per day, and we continue with 3 to 5 days in a row. So about 6 fish we sample to taken after the last treatment on days 0, 3, 6, 12, 24, and etc. to 48. Thank you. Next. For the histopathology analysis, data analysis was carried out on several organs, such as liver, spleen, kidney, and muscle. Based on the result of diagnosis, for the conclusion are drawn as follows. The antibiotics are considered safe or positive safe test if the antibiotic don't produce pathogenical change that the pathognomic when compared to the control group. In B, antibiotics are considered unsafe or negative safe test if the administration of antibiotic cause pathognomic pathological change. Next. Water quality analysis, we examine of water quality parameters such as dissolved oxygen, acidity, temperature 2 times per day until the during of study, and while for nitrate parameters are carried out at the beginning of the test. Next. This is the results. Before we treat the fish in the determination of withdrawal time of enterofloxacin, we check the enterofloxacin residue in the ordinary pellet fish and the pellet with the enterofloxacin and the goldfish meat. So we don't have to bias data. So there is a residue enterofloxacin in the pre-treated goldfish. Maybe this is we don't know about the record from the seed or the fry of goldfish that we collect from the fish farmer. So that's the data for enterofloxacin pre-treatment. So in the table 2, we collect the data according to the data, the day 0 to 24, enterofloxacin residue in the 2 group control and treatment coming decrease day after day until the end of the study. So look at the table 2. In the day 0, control group have 1.88 microgram per kilogram, but the treatment group have 156.19. So the antibiotic is already in the fish flesh. Yeah. Next. This is the data of the termination of withdrawal time. If we plot the data of linear graphic in the figure 1, we saw that if we equation for y for the 1 mRL in transport logarithmic for 100 or 100 ppb dose of MRL, we convert to the logarithmic is around 2 in the log concentration in the y-axis, and for the x-axis is a day that we have observed the withdrawal time of enterofloxacin. So in table 3 we have the same equation. For 1 mRL, we have 2 days for withdrawal time, and yes, pardon.
We are out time. Please conclude and close your presentation.
Okay.
Thank you.
On next. Conclusion. The termination for enterofloxacin withdrawal time for 1.5 mRL and 1 mRL for 2 days and 8 days respectively. The survival rate of gold in post challenge with iron-monocytic hydropolia for the positive control group and efficacy group were 86.7 and 88.9% respectively, and were not significantly different. For the result of histopathological observation on the liver, spleen, kidney, and muscle of carp, so no significant pathological change between the safety test, dose treatment, and the control treatment. And the value of water quality parameters for goldfish rearing media for 4 weeks. So a range that will still feasible for fish rearing activity in Fanta. Thank you.
Thank you, Mr. Manja, for the presentation. Close for okay, for the second presenter is Mr. Romi Sukarno.
Mas Rominda.
Thank you. Oh, sorry. This is not Romi. Sorry.
Sorry. For the second presenter is Mrs. Maya Neyana. I'm sorry, Mr. Romi. Okay, for Mrs. Maya, time is yours.
Yes, thank you. Thank you for the time. Good afternoon, everyone. I will begin showing you this slide. My title paper is Growth of Echumacotene Seaweed from Tissue Culture with Different Acclimatization in Hurun Bay. Next. The success of seaweed culture is inseparable from several factors such as the environment, seed quality, the method used, the availability of nutrient, and the initial density or weight in maintenance. The application of tissue culture technique to seaweed appeared in the 1970s to respond to market needs and seaweed culture practice. Tissue culture is a method of isolating parts of plant in seaweed. It is stylus and growing them under aseptic condition so that this part can multiply and regenerate into complete plant again like their parents. Next. The culture environment is the result of interaction between plant material, culture container, and the external environment of the culture space, having a very large influence on a tissue culture system. Several studies on tissue culture method on Echumacotene have been carried out, including using growth regulator, ZPT, various types of fertilizers, photoperiod, and different containers. Next. Purpose. The study was conducted to determine the effect of growth of Echumacotene seaweed from tissue culture at bamboo floating reef in the sea that had been acclimatized to different times in a greenhouse laboratory. The research is expected to be used as a reference material in producing good and sustainable tissue culture seaweed sailing from a greenhouse laboratory scale to a marine aquaculture scale. Next. This is the research location. Picture 1. This is map Hurun Bay. Hurun Bay where research location to use. Research method was carried out for 6 weeks in the greenhouse laboratory and 1 month at bamboo floating reef of the sea in Hurun Bay. Next. Picture 1 show tissue culture handling acclimatized in greenhouse, and picture 2 reread tissue culture handling acclimatized result in the sea. Next. Material and methods. Using AL randomized design, 3 treatment and 3 replication. Treatment 1, 2 weeks acclimatization. Treatment 2, 4 week acclimatization. Treatment 3, 6 week acclimatization. Talus was reread different time acclimatization treatment in greenhouse were reread at bamboo floating reef in the sea for 1 month. This is a formula in research. Next. The result. Table 1, absolute growth rate, daily growth rate, DGR, weight gain, and total harvest of HML seaweed from tissue culture with different acclimatization in Hurun Bay. From an average initial weight of 5 gram, at the end of the study, a different weight gain was obtained. Next. Figure 2, a graphic of talus weight growth of tissue culture sanding that have been acclimatized and reared on floating raft at the sea. Where the red line is the 4-week treatment showing the best talus growth. Next. The result, figure 3, graphic of daily growth rate of acclimatized tissue culture sanding for 2 weeks, 4 weeks, and 6 weeks in the greenhouse. Figure 3, where 4-week treatment showing at the best daily growth rate in the greenhouse. Next, the result, figure 4, graphic of the daily growth rate of tissue culture sanding that have been acclimatized and reared on floating raft at the sea. Figure 4, where 4-week treatment showing is the best daily growth rate in floating raft at the sea. Next. Table 2 is water quality in the rearing of seaweed sanding with different acclimatization. Where all parameters are still within the quality standard, but the nitrate parameter, above the quality standard, but still good enough for seaweed to grow. Next. Discussion. The 4-week treatment had the best weight gain, 30.5 gram, with no significant difference, P greater than 0.05 from treatment 2 week, 29.2 gram, but a significant difference, P less than 0.05 from treatment 6 week, 23.6 gram. The same trend was seen in the absolute growth rate parameter, which indicated a significant difference, P less than 0.05 between treatment 6 week, 0.84 gram, with treatment 2 week, 1.04 gram, and 4 week, 1.09 gram, but no significant difference, P greater than 0.05 between 2-week treatment and 4-week treatment. Observation on daily growth rate tissue culture seaweed sanding of the research showed the same trend, where 4-week treatment showed the best value compared 2-week treatment and 6-week treatment. Next. Conclusion. Different acclimatization times had a very significant effect on the growth of absolute weight of HML cottony tissue culture seaweed reared in a greenhouse laboratory aquarium. The longer the acclimatization time, the lower the talus weight growth and daily growth rate. 4-week acclimatization in the acclimatization that give the best growth of talus weight and daily growth rate. Next. Next. Thank you very much.
Yeah, thank you for Mrs. Maya. Please close for Mrs. Maya. Okay. Okay, the first presenter is Mr. Romi Suprato, but now the presentation will be represented by Mr. Igusti Murah Permana. Okay. For Mr. Igusti Murah Permana, time is yours.
Baik. Terima kasih. Thank you, Miss moderator. I would like to say good afternoon, everyone. I hope we are fine and happy this afternoon. I will try to deliver to a presentation of Pak Romi because Pak Romi as author, he has the institutional task. So that's why I try to deliver, and I hope you all of you understand what I will present. So the title of the presentation is Genetic Variation of Barramundi, Latus calcarifer from Indonesian and Australian population using SSR. Next, please. So the task, the outline of our presentation, introduction, and then material and methods, result, and conclusion. Next, please. So we know the barramundi or kakap putih is a potential species in the world, also in Indonesia, and a broodstock highly depend on the genetic variation for natural broodstock, and also we increase demand of superior broodstock. What should we do? And then for that model or for that statement, we try to assess the genetic variation using SSR or microsatellite and during the selective breeding. So in this study, to find out the allelic variability to direct the selection of founding parent in order to stabilize a base population. It's mean important for barramundi, for selective breeding. Next, please. I will give you briefly about the material and method. We analysis 50 sample per each individual broodstock from Australia and also local from Madura, East Java. We use DNA extraction, PCR, and then using the 6 microsatellite loci. Next, please. This is the primer that we use. We collect the primer from the reference, and then we try to apply. Actually, not 6, but more. But I think this 6 is better than others primer. Next, please. And then the result, the growth performance, the growth parameter of the first initial length from Australia, 3.05 centimeter, and from Indonesian, barramundi 2.8 centimeter, and then mean final length 38 centimeter, little bit bigger, and then from Indonesian 37. How about the mean length gain? 35, and then from Indonesia also 35. Initial weight is 0.5 gram for Australia, and Indonesia 0.45 grams. And the mean final weight is 820 grams, less than 1 kilos, and from Indonesia 787 gram. The main weight is 820, and from Indonesian is 786. Next, please. So this one result of visualization of PCR product from the Indonesian population using LCAM 37. As we seen in the figure, they are a good band, and then we try to get the DNA synthetic and then send to deliver to sequence. Next, please. The comparison of allelic frequency from Australia and Indonesian population, as you see in the figure, there were 4 alleles found together from the two populations. I think it's mean 148 allele 1, and number 2 is 161, 261, and the last is 271. There are 6 privat allele presenting only in the Indonesian population: 135, 160, 171, 197, 248, and 268. Sorry. Whereas only 2 privat allele, 249 and 270, appear only in the Australian population. That means there are different population, different allele between Indonesian and Australian barramundi. The next, please. How about the allelic pattern of barramundi from Australia and Indonesia? As we seen in the figure, allelic pattern is of Indonesian population is higher than the Australian population, like with heterozygosity, and Indonesian population higher than Australia. So that's why I think because the Australian population is they are already a selected broodstock we get from Fremantle Australia. Next, please. Oh, sorry, I forgot the conclusion. The conclusion, I added the conclusion, the frequency of allele with the Australian population at locus 148, 161, and 261 is higher than Indonesian population, but at locus 271 is unique for Indonesian population. Generally, privat allele and heterozygosity number of Indonesian population higher than the Australian population. I think that's all my presentation. Thank you very much.
Thank you, Mr. Igustin Murah Permana, for the presentation. Give applause for Mr. Murah. Okay, now we continue for the third presenter. The third presenter is Mrs. Niketut Maha Setiawati. Okay, for Ms. Niketut Maha Setiawati, time is yours.
Okay. Good afternoon, ladies and gentlemen. Thank you for the time. Where is my PPT? I would like to present my paper with the title Polyculture of Sea Cucumber and Millfish in Control Tank. Next. For introduction, some of previous research on polyculture of sea cucumbers had been done between Holuturia scabra and tilapia, and then with red seaweed, with Lithopenaeus fanami and Haliotis squamata. However, there are no available reports of polyculture of Holuturia scabra with milkfish. In the study, there is an idea of regulating the milkfish farming environment with Holuturia scabra through polyculture. Next. Please, next. One of the problems in polyculture system is determining the most effective combination of fish species in utilizing the natural food available in the culture environment. In addition, the combination of the fish species must be able to live together without causing competition for food or space. The present study was performed to investigate the growth and survival of sea cucumber, Holuturia scabra, and malefish reared in polyculture system in control tanks. Next. Materials and method. We use the 12 polyethylene tanks with volume 500 liter. Rearing sea cucumber and for rearing sea cucumber and malefish was done in a flow-through water system. And sea cucumber and malefish were used in the study were derived from hatchery. The initial size of sea cucumber was 3.4 until 5 centimeter in total length and around 3 until 4.9 gram in weight, while the initial total length of malefish 19 until 22. And the experiment assess different density of malefish in polyculture of sea cucumber and malefish in control tanks. Treatment in the study were A, monoculture of sea cucumber with 6 sea cucumber per tank, and B, polyculture of sea cucumber 6 and 2 malefish per tank, and C, polyculture with 6 sea cucumber with 4 malefish, and D, monoculture of malefish. Each culture which replicated in 3 tanks, 3 times. Okay. Next, the parameter measured in the study were the growth of sea cucumber and malefish and specific growth rate, the number of phases in treatment, total N, total P, and in the phases, as well as in the malefish and sea cucumber feeds. Water quality were included temperature, pH, salinity, ammonia, and light intensity. We collected every day from all the replicated tank for its treatment. Next. And result, survival rate of sea cucumber and malefish after 30 days of experiment achieved 100%. The survival implied the sea cucumbers and malefish could be farmed in polyculture system. In the system, uneaten feed and feces of malefish were utilized by the sea cucumber, which in turn support the growth of sea cucumber. For the figure 1, a body weight of the sea cucumber at the initial and termination of this experiment. At the end of the experiment, sea cucumber rate with 4 malefish per tank in treatment C had a higher final body weight than in treatment A and B. This result indicate the monoculture of sea cucumber and polyculture of sea cucumber with only 2 malefish tanks had similar final body weight. Okay. Next. And the total length of the Sikakamba both in monoculture and polyculture with 2 and 4 milk fish per tank was similar. Next. This is the total length of milk fish at the initial and termination of this experiment at treatment B, C, and D around similar. Next. This is a specific growth rate of Sikakamba was the highest in polyculture with 4 milk fish with the SGR 1.9% per day in comparison with only 0.2 in polyculture and with 2 milk fish, and at treatment A only 0.5. Next. This is the amount of feces, so that the polyculture of Sikakamba with 4 milk fish at treatment C resulted in the higher amount of feces, while monoculture of milk fish showed the lowest number of feces. Although the number of milk fish in treatment D and C was the same. However, in treatment D, there was no Sikakamba that can utilize the feces, and therefore it tended to dilute in the water column. As a consequence, is the number of low. Whereas in C, the feces of milk fish had been utilized by the Sikakamba as their feed. Next. This is a total N and P in feces in treatment and milk fish and Sikakamba feed. In polyculture, the total N in polyculture can decrease of total N. But in polyculture, total P in polyculture more higher than monoculture. Next. Water quality in the rearing media during the experiment. Temperature around 28, pH around 8, and salinity 33 until 34, and ammonia in the treatment A lower than treatment B, C, and D. Okay. Next.
I'm sorry, Mr. Setiawati.
Next.
I'm sorry, Mr. Setiawati.
Next.
I'm sorry, Mr. Setiawati. Hello?
Hello?
The time, the time is out. Please conclude and close your presentation.
Conclusion. For the conclusion, polyculture of Sikakamba and milk pigs resulted in the same survival with either monoculture and Sikakamba or monoculture of milk pigs. The polyculture of Sikakamba with 4 milk fish per tank showed a higher final weight of Sikakamba as well as milk pigs than the other treatment. Okay, thank you.
Yeah, thank you, Mr. Setiawati. Please give applause for Mr. Setiawati. Okay, the next presenter is Mr. Igusti Murah Permani.
Baik, terima kasih. Good afternoon, everyone. So thank you for the time, Mbak Dewa Ayu. Before I start, I would like to introduce. My name is Agustin Permana. I belong to BRIN, or National Research and Innovation Agency, the small part of BRIN in Gondol, Bali. So now I'm gonna share our work or topic related on abalone. The title is Sperm Motility of Tropical Abalone, Haliotis squamata. That's the impact of individual selection. Next, please. First of all, I would like to begin by introducing the research on breeding selection based on phenotypic from natural offsprings already succeed in Imrave or Gondol, Bali to produce the filial 1 to filial 3 from 2011. The male parent involving in abalone spawning is crucial for developing the high quality of seed. The availability of high quality, a large amount of seed is essential for the success of abalone farming. So, and then the low quality of spermatozoa may contribute to the decrease in the hatchability of egg. There has been a lot of investigation about that and into the role of selection in spermatozoa and abalone. That's why I purpose of this experiment was to determine the structure of male offspring sperm and its progeny under the impact of individual selection. Next, please. So this is the simplified material and method. We select the wild broodstock. Wild broodstocks mean from the nature. Were taken from Pukutatan Beach, is from Jembrana Regency, Bali, approximately this what do you call it, the longitude and latitude you can see in slides, and then 25 male and female from they had we spawn them and reach, if they reach the gonad maturity TKG3 and had the cell length between 6.5 and 7 cm, they were we used to spawning. This is the figure of gonad and then male, female, and then process of the spawning of abalone cell. Next. This is a protocol of individual selection. We built the protocol of individual selection from Research and Development Center Gondol, Bali, 2010, and then a cut off of 50% cut is performed after the 8th month, and then 5 to 10% selection after the 8th month to carry it out to separate male and female, and after that, if they reach the 6 to 80 month, the F1 will become broodstock and ready to spawning. The same individual selection technique method is used to create F2 and F3 and so on. Next, please. This is an individual selection diagram based on cell length and cell width and also weight. You can see in the slide. Next, please. And then the sperm. Next, please.
Recording in progress.
And then the sperm collection. Male abalone with back, please. Male abalone with the natural gonad were subjected under a temperature shock, as I mentioned in the front. and with the dry up and pure oxygen, the sperm that was released after the spawning then mixed with physiological sodium solution and added to the sperm cuvette. Sperm collection and in microtube is in 5 microliter in Leja chamber of sperm motility to count the sperm motility and viability. The motility level test will be carried out using the integrated with sperm class analysis. Next, please. Then the other parameter also we try to collect the egg diameter, degree of fertilization rate, and also hatching rate. A total of 50 eggs per 3 batch were measured in 3 other parameters like egg diameter, degree of fertilization rate, and hatching rate. The observation were carried out using a Nikon SMZ 1000 and Nikon Eclipse, also microscope coupled to a Nikon DSLR camera. Next, please. Data analysis, we analysis with rapid velocity, motility, and also the level test with microscope, SCA, and then other parameter we measure, and then a diameter, fertilization rate, and hatching rate, data obtained in ANOVA and correlation analysis with Excel program. Next, please. Then the result, I will give you briefly result. The average of the abalone, the average of spermatozoa abalone was 2.3 million cell per ml, with the highest of F0 or the wild of 7 million cell per milliliter. The highest progressive motil of the wild F0 of 89.8 and F1 80, F3 50, and F2 22. F0 and F1 were significantly different from F2 and F3. Next, please. This is the sperm velocity, also the greatest sperm velocity of the wild F0, and then yes, please.
We are running out of time. Please
Yeah.
And close your presentation. Thank you.
Yep. Next, please. This is the egg diameter, hatching rate, and degree of fertilization. Also, I found how the polyspermy penetration in squamata during the fertilization. That's in that condition. So next, conclusion. Next, please. Yep, the conclusion, a significant difference in sperm motility and rapid velocity was found between the wild F0 and other treatment in this experiment. This conclusive finding will help get insight into the effect of individual selection. The consequence of individual selection will aid in studying reproductive factor and sperm motility, which are most likely crucial in tropical abalone fertilization. Next, please. I think that's all my presentation, and thank you for your attention and listening. I'm going to bring time for the committee. Terima kasih.
Thank you, Mr. Mura. Give applause for Mr. Nicholas. Okay, now the last presenter in the session is Mrs. Nurhidayah. But for Mrs. Nurhidayah, we don't have your presentation. So please sharing your slide presentation and start your presentation. Thank you.
Thank you. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. Thank you for the opportunity given to me to convey the result of my research. The title, Exploring the Potential of Senility alba leaf boiling extract as Natural Preventive, measure against white spot syndrome virus in tiger shrimp fingerling. Mangrove, which are also known as mangrove forests economy that arises of biodiversity. This is mangrove, plant group brackish water between land and sea and tropical and subtropical region around the world, apart from providing protection and habit for forest species. Mangrove also have an important role in maintaining the balance of coastal ecosystem. One of important benefits of the mangrove ecosystem is ability for fight disease attached on tiger shrimp, especially the white spot syndrome virus. This is very determinant to the shrimp industry, causing a significant reduction in production and economic losses. This is indirect help reduce the risk of waste extraction because waste spread more easily in and steady environmental condition. The use of mangrove extract for the boiling Senility alba has become the concern in developing strategy for controlling shrimp disease natural. Senility alba, known for its bioactive compound, which have the potential for against pathogen include YSSV. One of the methods used utilizing Senility alba to produce extract from the result of boiling the leaves of the parts. We will explore the utilization of the boiled extract Senility alba to produce immunity for shrimp against YSSV attached. One of the disease prevention procedure in recommended the administration of immunostimulant to increase the resistance of natural immunity of shrimp seed to disease. The use natural material, the range from mangrove and their associated with disease control, the fishery sector has been top is limit to laboratory scale, including antiviral. The study aims to prevent YSSV disease in fingerling, tiger shrimp, through the use Senility alba mangrove herbal extract by immersion. Material and method. Water preparation, sterilization of water glass and aerasi equipment with caporate, neutralisasi natrium sulfat, animal acclimatasi salitas 28, preparasi Senility alba mangrove extract, leaf preparation mangrove Senility alba, drying, and then weight Senility alba 300 gram, add water 5 liters, boiling, and then Senility alba extract ready to use. Test animal. The study used test animal in the form of tiger fingerling post larva 20 individu per jar. 8 treatment were tried, show on the screen from A to F, starting from 12 hours of immersion with a time span of 12 hours each for treatment. NG positive control infection with YSSV but with no mangrove extract, and H negative control without YSSV infection and mangrove extract. Result, stage instalasi aeration equipment and a label, filling a sterile water and test animal research container, immersion of test animal with 1% sanitary alba extract, and then extracting test 15 ppt 15 minutes. Was infection by immersion in glass jar, and then move the test animal to the Sanitas 25A. Observasi daily mortality, sampling of water quality, total organic matter, NH3N, and NO2N at the beginning and in the study. Data stream survival was analysis for various followed by the last significant different test and descriptive water quality. The survival fingerling tiger stream. This slide, based on the result of soaking Sonerte alba boiling extract and different times show of figure 1. The picture you can observation of on the 5th day after infection with SSV in shrimp that had with soaked for different times. Tiger prawn mortality occurred in treatments taking from 1.7 until 6.67. Survival rate fingerling decrease every day until the 8th day. The survival rate of fingerling in the immersion treatment range from 71.7 until 85%. This shows that the concentration of SSV use was very viral, and 1% mangrove extract Sonerte alba effective inhibiting SSV infection. The water quality, NH3N concentration at the start was 0.8 ppm. After the maintenance period was increased, the concentrations of NH3N treatment with different value. The concentration of nitrate contact increase relative small. Increase nitrate concentration still needs to be considered because it can have negative impact on the health of the shrimps. BOT, the rearing period of shrimp larva and elevation in bahan organic value significant high demand for oxygen in the water, like the decomposition of organic substance. Conclusion, shocking Sonerte alba extract for 36 hours profit to be most effective method in preventing of fingerling from dying due to SSV attached resulting in survival 85%. The utilization of 1% concentration deposition of Sonerte alba extract can be a highly effectively strategy to enhance the productivity of shrimp farming and minimize the vulnerability of shrimp to SSV extract. Thank you.
Thank you for the presentation for Mrs. Nurhidayah. Please give applause for Mrs. Nurhidayah. Now we continue to the discussion session. Yeah. Please your head. Mention your name, institution, and mention to whom your presentation question is for.
Okay, first of all, I would like to introduce myself. My name is Justin. I'm one of the college students in the Faculty of Marine and Fisheries at University. I would like to ask you a question to Mr. Murad. From your presentation, I saw that your research using a complex method, like collecting the sample and measuring the parameters and others. I want to ask you a question. What are the difficulties that you have encountered or found during your research time? Thank you.
Thank you for Mr. Justin for the question. For the second question from please deliver your question.
Thank you. I'm Diana Wahatika, postgraduate student from Bogor Agricultural Institute. I would like to deliver my question to Mrs. Nurhidayah. What is the reason why you choose Soneratia alba for your research? And on your slides, you mentioned that Soneratia had bioactive compounds. Can you mention what is the bioactive compounds that contain in it as a natural preventive against WSSV? Thank you.
Okay, thank you, Miss Diana, for the question. And the third question for the session, please. Okay. We have a question from chat, from Mr. Haranto to Mr. Manja. The question is, from your table show that in fish, higher enflosatin compared to untreated fish, what does it mean in related to MRL? Okay. For the first,
I think the question already answered by Pak Manja, Bu.
Oh. Okay, thank you. For the first question from Mr. Justin, for Mr. Pura, please answer the question. Time is yours. Mr. Pura.
Sorry, Bu, I can hear clearly about the sound not so clear. Sorry.
Yeah.
About the questions.
Mr. Justin,
Sorry.
replay the question.
Okay, maybe I will repeat my question. From your presentation, I saw that your research using a complex method, such as collecting the sample and measure the parameters and others. So the question is, what are the difficulties that you encounter or found during your research study? Thank you.
Yeah, terima kasih. I think this is a very good question. The first, we did this research around 5 years, because we did the parallel research to collect F0 from the wild, and then selected with we are doing the selective breeding with individual selection. But unfortunately, I think for my research, they are not clearly succeed with that. If we are doing the individual selection for abalone, I think it's better we are doing the family selection or hybridization like that. I think, and then the parameter that I try to find out why the sperm hatching rate lower. The lower parallel with the generation like that. And then I try to get data from each batch spawning, F1, F0, F1, F2 and F3. I think that's all. I hope you understood what I mean. Thank you very much for the question.
Okay, thank you, Mr. Murad, for the answer. For the second question from Miss Udayana, for Mrs. Nur Hidayah, may respond to the question.
Thank you so much for your question. The reason why we choose Sonoratia alba in this research, because based on results of research by Sad and Murada, that Sonoratia alba mangroves produce antimicrobial activity against cartel microorganism and hypochlemic activity. So, as the use modified glucose increase serum insulin level and decrease blood glucose level. And the answer for the second, the bioactive compound that contain is Sonoratia alba, such alkaloids, steroid, saponin, flavonoid, phenolic, tannin. Thank you.
Thank you, Mrs. Nur Hidayah, for the answer. Okay, I'm sorry, the discussion time is over. You may still have further discussion with presenter during the break session outside the room, or you can chat privately. Okay. Now, we finally come to the end of our session. On behalf of the committee, I would like to thank the presenter for the insight and input from all participants during the session. Please give a round of applause for all of us. Thank you for the great work, and I will see you in the next event. And now, please back to main hall for joining the invited speaker from Professor Nurul Huda from Universitas Malaysia Sabah, and from Professor Helen Pang from Rostock University, Germany. Thank you.